Well, I'm touched and honored, and I thank the generosity of the Foundation and NWRI, and Joan I'll have to talk to, who wrote that letter, because now I know who it is. So I'm going to start by talking about the theme I've chosen: a biologist to engineers and engineer to biologists. And I'll talk a bit about my intellectual history and how I came to do what I've been doing. Just a reminder of history of what we did before QMRA, and challenges and future problems both with QMRA and in other fields that impinge on the intersection of my interests. And then I'll finish up with a discussion of what this means as an educator. How do we educate the next generations? So, before I chose my theme, I read through a lot of the lectures that are on the website. And I was intrigued by the inaugural recipient, Bruce Rittman, who I wish was here, who in his lecture talked about two kingdoms of water science and water technology. And so, in some sense, what I'm going to do is expand on that, because we have many more kingdoms now than just that. And then the other basis for my talk is how I recall my late Ph.D. advisor, Dick Engelbrecht, always describing himself. And Vern will correct me if I've got the quote wrong, but I always remember Dick used to describe himself as a biologist to engineers and an engineer to biologists. And so I think what I've done is at that interface, and I've established additional interfaces as well. So basically what I do is at the intersection of three circles: microbiology, the water environment, and health. I started out in biology, got my master's degree in environmental engineering. My freshman year coincided with Earth Day, so that was really an impetus to move into environmental work. And I've always been interested in quantitative problems. And then I had a lot of serendipity along the way that caused me to invent this field, and I'll talk about that a bit more. So this is the one good picture I have. Maybe prior to tonight, maybe we'll have another picture after tonight, guys, of Joan, Chuck Gerba, and I. So I did a fellowship at EPA in the mid-1980s, and it so happened that Chuck, who I never had met before, was in the same year of that fellowship class. And so we got to know each other well then. And at that time, Joan was finishing up her graduate work at Arizona. And so after the fellowship year, I came to visit Chuck, and I met Joan. And a few years later, she did a fellowship at EPA. And we really bonded ever since. Yeah, 30 years. It's frightening, Joan. So what is QMRA? Just a very, very quick overview. We identify hazards that microorganisms pose. We look at their dose response to people. What's the relationship between dose and likelihood of having a bad effect? We quantify the exposure. We characterize risk. And then we work with risk managers. Many of the utilities in this room are risk managers at developing interventions. Now, QMRA really became important when we started to be able to measure pathogens in water with some sense of reliability, and when we realized that there was a trade-off between treatment we interposed to reduce pathogens and adverse side effects that could result, for example, by increasing disinfection dose. Now, I want to remind you what we did before QMRA. Actually, the first written reference to a disinfection dose that I've seen was by Chester Averell in 1832. And he was a professor of, and you'll get a kick at this, chemistry and classics at Union College in Schenectady, New York. And Schenectady was in the middle of a disease outbreak, probably cholera, in the summer of 1832. And he wrote a published letter to the mayor of Schenectady giving disinfection guidelines. If you work out the math and you make some assumptions, treatment technology of 5 grams per liter. Probably kill everything. I'm sure it worked. But this is 40 to 50 years before the formulation of the germ theory of disease, and probably 60 or 70 years before the germ theory was really widely accepted. And so now I'll wear my engineering hat. Sometimes we wind up doing the right thing for, I wouldn't say the wrong reason, but for a reason that we don't fully understand. We then fast forward about 80 years. Where did the Coliform Standards come from? As many may know, the first Coliform Standards were set by the Department of the Treasury to regulate water quality in railroads. And they had drinking cups on the railroads. And I have been able to find a contemporaneous picture of the drinking cup, but it was probably about the same capacity as the modern 4-ounce cup now. And they were measuring bacteria by the MPN technique, which is very imprecise. And you could detect maybe 2 coliform with the standard 5-2 MPN test in the capacity of this drinking cup, which is roughly 100 ml. How many of you still have a 2 coliform standard per 100 ml today? I won't embarrass anybody to raise their hands. Yeah, that's still the state number, right? That's where it comes from. It worked. It worked. Okay, so, this is, it happens to be from Philadelphia, but a number of other cities have graphs like this that show that first as water filtration was installed in Philadelphia in 1906, and then water chlorination was installed in 1913. The typhoid case rate dropped by 2 orders of magnitude over that interval. It worked. We no longer have typhoid. Well, maybe there's 1 or 2 cases in Philadelphia a year, but it's at the noise level. So then to 1991. Viruses were a concern because Chuck Gerber and many other people established that viruses could be more resistant than coliform. We had Giardia outbreaks occurring. And so Joan and Chuck and I and Stig Wrigley, who I'm delighted that NWRI got a letter from, really had the impetus to say, well, what could we do to develop a standard for Giardia and viruses? And so we applied QMRA, which, as Joan said, I first developed in 1983 for a wastewater issue in Chicago. And we came up with the concept of log reduction as a function of influent water quality. So how many utilities hate LRVs? Well, blame me, okay? So, we came up with that with the idea that we'd protect down to a level of 1 in 10,000. I could give another total lecture on the 1 in 10,000. It was a heuristic at the time. It was probably wrong. We didn't have a better way of doing it. There were roughly 25,000 cases of waterborne illness and detectable outbreaks from CDC at the time. The population was about 250 million. Divide one by the other, you get 1 in 10,000. It ain't rocket science. But unfortunately, it's been codified. And I really hate this. Now, I know that, coming to California, I've spent all my life in the Northeast and the Midwest. What the heck do I know about water reuse? So let me just tell you. Vern probably knows the top map. I actually did a piece of my Ph.D. work on the Salt Fork of the Vermilion River, which goes between a small wastewater treatment plant and a small drinking water treatment plant. Sampling through the wastewater plant, sampling that stream reach, and sampling in the water treatment plant. I don't think we called it reuse at the time, but it was certainly reuse. Then I moved to Chicago. And here's really where I came up with QMRA. At that time, the other gray hair and gray beards in the audience may remember the first secondary wastewater treatment plant standards required wastewater disinfection. And then EPA went ahead and removed that requirement federally. And Chicago is very interested, as a major wastewater utility, of stopping chlorination. Now, about 90 stream miles downstream of Chicago is Peoria. And the wastewater flow in Peoria, the majority of which is, the drinking water intake at Peoria from the Illinois River, the majority is wastewater from Chicago. And so American Water, which owned the utility in Peoria, contacted me and said, what could we do? And so, based on a consulting project for American, I invented the field. But that's wastewater reuse as well. So fast forward, I wound up being on the first NRC panel that looked at issues in potable reuse. I'm sorry Jim Crook isn't here tonight. I'll mention it to him. Those of us that were on the panel, including Jim and myself and Rhodes, in retrospect, regret having had the phrase sneak in on us that potable reuse should be the option of last resort. And so that, I think, set back the field, particularly here in California, by some period of time. And then we had a subsequent study 10 or 12 years later, the second NRC report, where I think we came to the realization that what we said was probably wrong, and I'm glad to see that things are proceeding at a better pace now with the expert panel from NWRI and the state actions that are being taken to enhance the option of potable reuse. So, a couple months ago, I was at the IWA Reuse Conference in Long Beach, and I was very taken by one of the discussions that occurred after one of the papers. And I think, and I put this in my written text, and I think I'm happy being quoted on this, the modifying words of deliberate, unintentional, direct, indirect are really no longer useful. The time has passed. And we need to instead look at the quality of the water in the context of the use that it's being proposed for, and develop treatment requirements and management options that are fit for purpose. That's the new paradigm. Now, I want to talk about emerging and future problems at the interface. And actually, if you can see the background image, many of you that are familiar with paving stones know that the weeds grow up at the interfaces. And so what I would submit to you is the interesting problems and the interesting science and engineering that needs to be developed are at the interfaces between different areas of study. And I want to talk about 4 of them that I see, and I'll move to the issue of education. So we've now been moving from the use of indicators, the 1914 Coliform Standard, to molecular biology. First with quantitative polymerase chain reaction, essentially photocopying DNA from target organisms and measuring it. It's going to be more voluminous in terms of data when we get to sequencing. But we don't really have a good understanding of how we connect that information to human health risk. And I'll give a couple of examples. So, we recently did a project with American Water My recent Ph.D. student, Carrie Hamilton, is in the audience, and she did a bit of this work for her doctoral dissertation. And this is just a piece of it where we looked at Legionella in 19 utilities that are treating wastewater for reuse around the country. And we used 3 different methods, or I should say American, who did the lab work, used 3 different methods. Culture techniques for Legionella, qPCR techniques for Legionella, and a modification of qPCR called EMA qPCR, which attempts to differentiate between what's thought to be the viable component and simply the DNA. And the numbers, particularly comparing culture to the other methods, are very different. What does this mean? Do we need to worry about non-culturable Legionella DNA? Don't know. I don't have the answer to that. It needs further study and further research. Now, when we get to sequencing, the issue is even more difficult. With qPCR, at least you start out with a known sequence of DNA and see whether or not that sequence is present for the particular organism and what you're measuring. With direct sequencing, you take an entire sample, all the water from this glass, and look at all the DNA that I can amplify. And using that method, then match up the DNA sequences that you see with known libraries of DNA sequences from a variety of organisms. And so this, again, is work from Carrie that she did with colleagues in Brisbane, Australia at CSIRO. Well, you have to look at the colors. Don't bother reading the legends. We see lots of different things in different relative amounts. Each of the colors are a different class of bacteria. With sequencing, you don't get absolute numbers. You get relative proportions. We have no way to connect that to risk, let alone the issue of viability and infectivity. So it sort of reminds me, and I'm stepping in dangerous waters because many of the prior laureates and the other guests know more about chemical pollutant measurement than I do. But it reminds me of the early days when I was a student where people were starting to use gas chromatography, mass spec, on various water samples and would get a lot of traces and a lot of peaks without having the information or the knowledge to interpret that and connect that to human health risk. We're at a similar stage now with direct sequencing of environmental samples. Second big issue, emerging pathogens. What do we do beyond CGV, Cryptosporidium Giardia viruses? So probably some of the utilities are saying, why should I give the state any bright ideas? We know there are other problems out there. Over the years, I've looked a lot about Legionella. Nikki Eschbolt in Alberta has coined the term saprozoic pathogens. Pathogens that are capable of growing in the water environment under ambient conditions. Legionella is one example of that. You may have non-detect Legionella leaving a water treatment plant, but when it gets to a cooling tower, when it gets to an air conditioner, when it gets to an ornamental fountain, when it gets to a distribution system, it might amplify. We know that these organisms are increasing in importance. This is the CDC time history of waterborne outbreaks. Starting from the 70s until 2012 is the last time point on that graph. All you need to see is that we no longer have the grays and the blues and the whites, but in recent years we have a lot of blacks. And the blacks are bacterial disease, the biggest chunk is Legionella, and then non-Legionella bacterial disease. So those are emerging as significant problems in reported waterborne outbreaks. And I give the caveat that reported waterborne outbreaks are only a small fraction of the total waterborne diseases that we face. If you look at Legionella, the case rate is increasing dramatically. This is the CDC data from all sources, not just water, but for Legionella, the vast bulk of those have their origin in water in some way, shape, or form. Since 2000, the case rate has more than tripled. So the cases are on the upswing. There are other organisms out there that are saprozoic. Mycobacterium, non-tuberculosis mycobacterium, not the TB that your grandfather passed away from, but the non-tuberculosis. 90,000 cases in the US estimated per year. We don't know what fraction of those are water, but some fraction, because Mycobacterium can be found in water. That's been widely established. They can amplify through similar approaches as Legionella. Campylobacter can also amplify. There are about 2 million cases per year in the US, probably mostly food, but some fraction are water. And in the US, we seem to do a poor job at diagnosing and detecting Campylobacter in human cases. And the last, which we know the least about, are pathogenic Pseudomonas. There's some fraction of the population that appear to be highly susceptible to pathogenic Pseudomonas. There are probably others that we don't understand and comprehend yet. So the public health burden is out there. Third broad theme I'll present to you is antibiotic resistance. So the first antibiotics were discovered and used in the late 1920s, early 1930s, penicillin. This figure is out of the UK, but the time bubbles don't differ that much between the UK and the US. Within 12 years after the first discovery of penicillin, the first resistant bacteria to penicillin was detected, the blue circle. And then you see, as the other classes of antibiotics were devised, the rate at which antibiotic resistance is found appears to become even more rapid. Some people have, I'm not sure I'm this pessimistic, but some people have said we're at the end of the age of antibiotics. It's a major public health problem. We don't know how it develops in a quantitative sense. We know that if you go out to the environment, you can measure antibiotic-resistant genes and antibiotic-resistant organisms in people, in animals, in the environment, including in some sludges, in some wastewater, and even in some reclaimed water. What does that mean? We don't know, because we really need to understand the various rates. How rapidly do things move between those compartments? And what would happen if, for example, we got better controls in the water sector? Would it make a difference on public health, or would it not? Can't answer that question now. I know there are a variety of groups in the US and the EU that are studying this, but a lot more work is going to be needed to understand whether or not water scientists and engineers have an important role to play in reducing this health burden. Fourth topic is climate. Yes, the climate is changing. Yes, it affects hydrology, rainfall, droughts, floods, etc. But it also affects pathogens. It will affect the rate at which they grow, the rate at which they run off from surfaces and from animal wastes put on the environment, and it will impact water quality. We did a study getting back to Legionella about 12 years ago with a colleague of mine in our public health school, looking at Legionella cases in the metropolitan Philadelphia area. And essentially, we got good correlation between the Legionellosis case rates and the humidity and precipitation. not temperature. Summer cases were really driven by humidity and precipitation. This finding has now been replicated at other places around the world. With climate change, with increase in rainfall, with increase in humidity, bingo, what's going to happen is Legionellosis cases will increase as well. Climate will increase the presence and growth of pathogens. It will increase the hosts for microorganisms, whether they be amoeba that cause the saprozoic organisms to multiply, or insect vectors that may transmit infectious agents. It will cause increased runoff, increased fluxes of pathogens to water bodies. Will the impacts, particularly of increasing pathogen loads, be significant enough to necessitate greater log reduction values? So that's a big picture. LADP, Metropolitan, the other major utilities hopefully are starting to think about that. And last theme, I pulled the slide from one of your papers. The sustainability and health intersections. So there's a lot of talk, especially on the wastewater side of the business, about redesigning wastewater utilities to be net zero energy, net zero discharge, some combination of the two. And a lot of flowcharts in the literature and a lot of ideas have been presented. But I want to submit to you that there's a third leg of the triangle that hasn't been totally explored yet. Yes, we want to reduce energy consumption. Yes, we want to reduce discharge of effluent. Yes, we want to get improved materials recovery. But there's a human and ecological health dimension associated with what processes are used to get there. And we're not going to be able to achieve everything ideally. So we need to assess what the balance point is between reducing the net energy cost, improving materials and water recovery, and reducing human and ecological health impacts. And so that remains an engineering and public health challenge. And obviously, of course, cost enters into this as well. Given enough money, you can solve everything, but how many utilities here have enough money? So a brief word of where QMRA is going, and then I'll finish up with education. We have a broad spectrum of pathogens that we haven't really totally considered. Helminths are one, per our conversation earlier, and fungi are another. And fungi are interesting because they may also increase in importance with climate change. And so we need better models and better data to formulate those models. We need to integrate them with disease transmission models. How do we predict outbreaks? Host and pathogen physiological state, and then finally repeated exposures. I want to finish up with education. That's a photo of the main building at Drexel, which was founded in 1891 and still in use. So I've been at 3 different universities, and in my first university position, I had a dean who told me, and this was in 1978 or 79, that in 25 years we wouldn't need environmental engineers because all the problems would be solved. And so I just put together a word cloud of some of the hot topics that have emerged in that interval. What we can hope to do is to educate students at a rapid enough pace to keep up with the new problems that are emerging. I just heard during the reception lithium is emerging as a problem here in California. I need to look into that. That's intriguing to me. So in thinking about it, one of the things I recalled was that James Morgan, who was an early recipient of the Clarke Prize, gave a lecture at the National Academies for the Wallman Award. And Jim used the concept of the stream. In his lecture, he talked about the evolution of water and wastewater treatment processes. So I've kept his stream, but I wanted to use it instead to talk about the stream of water knowledge. And we've integrated many disciplines since the turn of the 19th century into what we need to know to solve water problems. From just being concerned with hydraulics and geography, incorporating public health, incorporating microbiology, meteorology, economics, analytical chemistry, and so on and so on and so forth. And so a couple things I want to drive home with that graph. First of all, we continue to be influenced by new tributaries. But also note that the number of things I put down at the far end of that stream are increasing in the rate at which they're entering what we need to know. If you remember back to Alvin Toffler in the 70s, I guess, talking about future shock. Future shock is because the future is moving at an increasingly rapid rate. We're incorporating more and more streams of knowledge into what we need to know to solve problems. One person can't do it all. And so in education, people are talking about producing T-shaped professionals, where somebody has a significant expertise in one particular narrow area, but enough of a breadth to understand and communicate with other professionals who go deep in other areas. And of course, if I were enough of a structural engineer, I could say, well, if I put those T's together and bolt them, I'll get a pretty strong structure. And so that's the concept. Everybody doesn't need to know everything, but if you get enough diversity in what people know and they have enough ability to crosstalk with each other, problems could be solved in a better way than they have been. And one particular challenge educators have is a lot of the restrictions on education, especially in the engineering sector, focus on knowing people who know a lot about a narrow field, but not enough talk about breadth. And so my plea to practitioners is push back. If you're on advisory boards or deal with universities, students need to have breadth to be able to communicate with other professionals in related areas. So I want to finish up with a quote from Louis Pasteur, and I happen to share a birth date with Louis Pasteur. In the field of observation, chance favors the prepared mind. I put the original French there for those who might want to read it in the original language. Our challenge is preparing minds for the future. As educators, as researchers, maybe even as managers and supervisors. A lot of acknowledgements. First of all, family. My wife, who couldn't be here but is with us in spirit from 2,400 miles away. My late parents. I had two cousins who were remarkable scientists and MDs, Murray and Norman Jarvik, that really got me thinking about science at an early age. Dick Engelbrecht, Jim Patterson, who were my formal mentors. Vern, who was on my doctoral committee. Roger Manier, who really got me interested in environmental engineering as a student at IIT at a very early stage. A number of funding agencies, Joan, Chuck, West Pipes, many students, including Kerry Hamilton is in the audience, and so forth. Mark Weir, who wrote that letter that Joan read from, who's now a professor at Ohio State. A number of key practitioner colleagues, Rhodes, Cecil Luhing, Marc Le Chevalier in particular. One last acknowledgement. The Irvine family and the Joan Irvine Smith and Athalie R. Clarke Foundation, NWRI, Joan Rose and Joe Hughes as my nominators. And I put this image up. I did reading on the Irvine family history, and lo and behold, I discovered not only did I share a birth date with Louis Pasteur, but James Irvine I had the same birthday. So thank you very much.